Welcome back to the Deep Dive, the show where we take your sources and turn them into a comprehensive understanding. We filter out the noise to give you the crucial nuggets of knowledge you need to be truly well-informed, with maybe a few aha moments along the way. Today we're tackling a topic that is absolutely fundamental to, well, literally processing everything we put into our bodies. It's a hidden world of internal chemistry and sophisticated protection. We've got a stack of detailed source material here, diving deep into the how and why of it all.
Indeed. The source material you've shared provides a truly fascinating look into gastrointestinal secretions, this complex mix of fluids, enzymes, mucus, other substances produced all throughout the digestive tract. It's a real deep dive into the cellular and physiological mechanisms that make digestion possible, but also, critically, ensure the gut doesn't digest itself.
Exactly. And our mission today is to take this wealth of information and really unpack it for you. We're going to explore every corner of this text, focusing on every topic, every concept, so you can feel like you've truly mastered the basics of how our gut produces exactly what it needs, when it needs it, incredibly precise. Think of this as your essential guide to understanding the secretory engine of the alimentary tract.
We'll explore the different types of glands involved, what they secrete, where those secretions go, what vital jobs they perform, from breaking down food to providing a protective barrier, and how their production is so precisely controlled moment to moment through a complex interplay of nerves and hormones. It's a system of remarkable complexity and coordination that operates continuously, adapting dynamically.
So get comfortable. Maybe grab a glass of, well, something that won't trigger too much acid reflux just yet.
Right.
Let's dive into the secretory functions of the alimentary tract. Okay, let's unpack this right from the top. The source material lays out that gastrointestinal secretions serve two absolutely primary overarching functions, essential for survival. What are those core jobs?
Right. At their most fundamental level, these secretions have two major roles. First, they provide the digestive enzymes. These are necessary to break down the complex molecules in our food proteins, carbs, fats into smaller absorbable units. This enzyme activity starts right in the mouth and continues all the way down to the end of the small intestine, the distal ileum. Second, and just as vital for the system's integrity, they secrete mucus. Mucus is produced from the very start, the mouth, all the way down to the very end, the genus. Its main job, providing essential lubrication and protection for the entire lining, preventing damage as food and waste move through.
So chemically dismantling food and physically protecting the pathway, keeping things moving smoothly, breaking down and building a shield. Makes perfect sense. The text also mentions something really key. The body isn't just constantly pumping out juice. The quantity and even the types of secretions are often tailored very specifically, right, to the food passing through. It's not a constant flood. It's highly regulated, adaptive.
Precisely. This is where the system's efficiency, its intelligence, really shines. The body secretes what's needed for the specific food components present. It senses, if there's a lot of fat or protein, and it secretes it exactly where it's needed along the tract, often in a composition optimized for the conditions in that particular segment. Enzyme secretion, for instance, ramps up significantly when food's detected, and the mix of enzymes, particularly from the pancreas and stomach, can shift depending on the meal's makeup. It's dynamic on demand.
The efficiency and precision built right in. Now, the sources describe several different types of glands involved. You mentioned simple cells, complex organs. Can you walk us through the structures making these secretions?
Certainly. We can think of this as a kind of hierarchy based on complexity and location. Starting at the simplest level, you have literally billions of single-cell mucus glands. Just individual cells scattered throughout the epithelium, the gut lining, mouth to anus. Because of their shape, they're often called mucus cells or goblet cells. Their function is incredibly direct, very localized. They respond instantly to any local irritation by extruding mucus directly onto the surface. Immediate lubrication, immediate protection.
Like tiny individual lubrication stations everywhere, ready on contact.
A very effective and widespread system, yes. Moving a step deeper into the gut wall, many areas aren't flat. They have pits. These are invaginations, like little tunnels of the epithelium extending down into the submucosa layer. In the small intestine, these pits are particularly deep, called crypts of Lieberkühn. These crypts contain specialized secretory cells, different from the surface cells. Figure 65 in the source shows one, gives a sense of their internal machinery.
Okay, surface single cells for immediate local stuff, then these deeper pits or crypts with specialized cells for more complex mixtures. What's next in complexity within the gut wall?
Third, in specific regions, mainly the stomach and the very beginning of the duodenum, we find large numbers of deep tubular glands. Unlike the simple pits, these are more complex, tube-like structures extending further down. The source shows a typical one in figures 65-4, an oxyntic gland from the stomach body. These deep tubular glands are specialized for secreting potent, specific substances. The oxyntic glands, for example, are the primary source of gastric acid and pepsinogen, the stomach's heavy hitters.
And finally, the big players, the accessory organs sitting outside the gut tube itself, connected by ducts.
Yes, the complex glands. Large accessory organs providing significant volumes of crucial secretions. This group includes the major salivary glands, parotid, submandibular, sublingual, plus the pancreas and the liver. These are typically compound acinous glands, like the salivary gland shown in figure 65-2. Lots of small secretory units, acini, draining into branching ducts. These glands lie outside the alimentary tract walls. Millions of acini and duct cells feeding secretions into ducts that empty into the gut. The liver's structure is unique, as the source notes, but it functions as a vital complex gland providing bile, essential for fats.
So this incredible spectrum of machinery, tiny cells to massive organs, a huge logistical operation. Now that we know what and where, how does the system get the signal to start secreting? The source details several basic mechanisms.
Well, the most direct stimulus is simply the presence of food, physical or chemical contact with the gut epithelium. This contact in a specific segment usually stimulates glands right there and maybe nearby areas to secrete moderate to large amounts of juice. Part of this is just the food directly touching and stimulating surface cells.
And this local activation isn't just direct touch, is it? The enteric nervous system, the gut's second brain in the wall, gets activated too. What kinds of stimuli does it respond to?
Exactly. The enteric nervous system is incredibly sensitive. The stimuli that strongly activate it, triggering more secretion, are, first, tactile stimulation from the food itself. Second, chemical irritation from food components or digestion byproducts. And third, distension, the stretching of the gut wall as it fills. The nervous reflexes generated by these local stimuli spread within the gut wall and increase secretion from both surface mucus cells and the deeper glands in that area.
What about the big nervous systems, the autonomic ones from the brain and spinal cord, parasympathetic and sympathetic? How much influence do they have?
A major role, especially the parasympathetic system. Parasympathetic stimulation almost always increases secretion rates. This effect is particularly strong in the upper tract glands: salivary, esophageal, gastric, pancreas, Brunner's glands in the duodenum, which get heavy input from the glossopharyngeal and vagus nerves. Parasympathetic input is also significant for some glands in the distal large intestine via pelvic nerves. For much of the small intestine and the start of the large intestine, it's more about local signals.
And sympathetic stimulation, the source calls it a dual effect. That sounds interesting.
It can be. It highlights this interplay between nerves and blood flow. Sympathetic stimulation alone might cause a slight increase in secretion from some local glands, a minor direct effect. However, sympathetic stimulation powerfully constricts the blood vessels supplying these glands, reduces blood flow significantly. So if copious secretion is already happening, driven by parasympathetic or hormones needing lots of blood supply, then adding sympathetic stimulation can actually reduce secretion, sometimes quite a bit, primarily because the blood supply, the raw materials, is cut back.
So a small direct boost versus a big cut in supplies. Less blood flow, less fuel. Really shows how critical blood supply is for high secretion rates.
Absolutely vital. Beyond nerves, the source highlights crucial regulation through hormones, particularly polypeptide hormones from the stomach and intestine.
How does that hormonal regulation work?
These hormones are released from specialized endocrine cells in the GI mucosa, responding to food in the lumen. Unlike fast, local nerve signals, these hormones enter the bloodstream, circulate, and bind to gland receptors elsewhere, stimulating secretion. This is especially important for boosting gastric and pancreatic juice output when food hits the stomach or duodenum. It coordinates a widespread response across organs.
So local senses, autonomic nerves with that dual sympathetic effect, and systemic hormones coordinating things. Lots of layers. Let's zoom in again right down to the cell. Figure 65-1 shows a typical glandular cell. How does one cell actually make and release these things?
Right. The source outlines this multi-step process. First, the cell needs fuel and building blocks, nutrients. These diffuse or are actively transported from blood capillaries into the base of the cell.
Needs supplies delivered constantly.
Exactly. Second, inside, often near the base, lots of mitochondria, the powerhouses. They use oxidative energy from those nutrients to make ATP, the cell's energy currency. Third, ATP energy plus substrates from nutrients fuels the synthesis of the organic secretory stuff, enzymes, glycoproteins. This happens mainly in the endoplasmic reticulum and Golgi complex. Ribosomes on the ER specifically make the protein parts.
Raw materials in, energy made, products assembled in the ER and Golgi. Then what?
Fourth, the materials move through the ER tubules to the Golgi. It takes about 20 minutes according to the source. The Golgi is usually near the apical end, facing the lumen where release happens. Fifth, in the Golgi, they're modified, concentrated, packaged into secretory vesicles, membrane-bound packets. These vesicles, full of concentrated product, are stored at the apical end, waiting near the exit.
And they wait for a signal. How do they actually get out?
They stay stored until nerve or hormonal signals arrive at the cell membrane. These signals bind receptors, triggering internal pathways. A common pathway increases the membrane's permeability to calcium ions. Calcium flows in from outside. This calcium influx is the critical trigger. It causes the vesicles near the apical membrane to fuse with that membrane. The fused membrane breaks open, releasing the vesicle contents outside. That's exocytosis, shown nicely in figure 65:1.
That calcium-driven fusion and release. A fundamental cellular mechanism.
It's a beautiful example of controlled release. The source also stresses that besides organics, cells need to secrete water and electrolytes as a vehicle. This fluid washes the organic stuff out. It's also driven by nerve stimulation and hormones, causing water and salts to pass rapidly through the cell.
Okay, we've got the machinery and cell controls. Let's focus on one of those primary functions. It's everywhere. What exactly is it, and why is it so important?
Mucus is this thick protective secretion, mostly water and electrolytes, but its key feature is a mix of several glycoproteins, large polysaccharide molecules linked to some protein. The exact mix might vary slightly, but its essential properties are consistent and incredibly beneficial for the gut lining.
And the source lists several crucial characteristics, 6 key ones.
Yes, 6 key properties. First, it's adherent, sticks tightly to food and, crucially, to the gut wall, forming a continuous film. Second, it has enough body viscosity to effectively coat the wall, a physical barrier preventing direct contact between food and the delicate epithelium.
Literally building a protective wall.
Precisely. Third, excellent lubrication. Low resistance to slippage. Allows food and later fecal matter to slide along easily, minimizing friction and damage. Fourth, especially in the large intestine, it helps fecal particles adhere to each other, forming solid feces for expulsion. Fifth, it's strongly resistant to digestion by the gut's own powerful enzymes. Absolutely essential. If pepsin or trypsin broke it down easily, it couldn't protect. And sixth, its glycoproteins have amphoteric properties. They can buffer small amounts of both acids and alkalis. And crucially, mucus often contains significant bicarbonate, HCO3. This specifically neutralizes acid contacting the surface. Extra chemical defense.
So not just slippery, it's sticky, tough, resistant, and buffering. A multi-talented shield.
Exactly. This combo allows smooth passage while preventing physical and chemical damage. The source's example about swallowing dry food without saliva, that difficulty really shows the power of mucus lubrication, even before you consider the deeper protection further down.
That example really hits home. Speaking of saliva, let's dive into that first major complex secretion. Which main glands are involved and how much do they make?
The principal salivary glands are 3 pairs: parotids near ears, submandibulars under jaw, and sublinguals under tongue, plus many tiny buccal glands in the mouth lining. Together, they produce a lot, 800 to 1,500 milliliters daily. Average around 1,000 milliliters a liter a day. Table 65:1 shows this.
A whole liter. And it's not just one type of fluid, right? Different protein components.
That's right. 2 major types of protein secretions from different cells. A serous secretion watery fluid containing ptyalin, which is an alpha amylase enzyme, starts digesting starches in the mouth, and a mucus secretion containing mucin, the glycoprotein, that provides lubrication and surface protection.
Do all glands make both, or do they specialize?
Some specialization. Parotids secrete almost entirely the serous type, mostly the enzyme. Submandibular and sublingual glands secrete a mix of both serous and mucus enzyme and lubricant. The tiny buccal glands secrete only mucus for local lubrication in cheeks and lips. The source also notes saliva's pH is usually 6.0 to 7.0, slightly acidic to neutral, good for ptyalin's action.
Okay, different glands, different contributions. What about the ions? Text says concentrations are quite different from plasma. How's that achieved?
Yes, very distinct. Saliva is notably high in potassium, K+, and bicarbonate, HCO3, much lower in sodium, Na+, and chloride, Cl, compared to plasma. This happens via a clever 2-stage secretion process shown for the submandibular gland in figure 65:2, representative of the major glands. Stage 1 happens in the Acini, the secretory cell clusters. They secrete a primary fluid with ptyalin, mucin, and ions similar to plasma. Then, as this primary fluid flows through the ducts, stage 2 occurs. Duct lining cells actively modify the ions. First, Na+ is actively pumped out of the duct lumen back into blood. Simultaneously, K+ is actively pumped into the duct lumen, exchanging for some Na+ einpass. More Na+ leaves than K+ enters, making the lumen electrically negative. This negative charge pulls negative Cl passively out of the lumen too, following the Na+ psi. So Na+ and Cl concentrations drop dramatically. Second, bicarbonate, HCO3, is secreted into the lumen by duct cells, partly passive exchange for Cl, maybe partly active secretion too.
So the ducts are like a processing plant, retuning the ion balance, pulling out sodium and chloride, adding potassium and bicarbonate.
Exactly. The net result at rest, slow flow, is low Na+ and Cl, about 17th to 110th of plasma, high K+, 7 times plasma, and high HCO3, 2-3 times plasma.
What happens during maximal salivation? Fast flow. Does that change the ions?
Yes, significantly. Acini can boost primary secretion up to 20-fold. With rapid flow, there's less time for ductal modification. So at high flow rates, Na+ and Cl concentrations are higher, maybe half to 2/3 of plasma. K+ concentration is lower, only about 4 times plasma. Still different, but less extreme.
Fascinating how composition changes with demand. Beyond digestion and lubrication, saliva is vital for oral health, right? The source emphasizes protection.
Absolutely critical. Under basal conditions, awake, there's a slow, continuous secretion, about 0.5 millimin, mostly mucus type. Crucial for mouth health. The mouth has tons of bacteria, some harmful. Saliva prevents tissue damage and tooth decay in several ways. First, the flow itself acts as a constant rinse, washes away bacteria and food particles they feed on, continuous cleansing. Second, it contains antibacterial factors, thiocyanate ions, proteolytic enzymes like lysozyme. Lysozyme attacks bacteria, helps thiocyanate enter bacteria to kill them, digests food particles. Third, saliva often contains significant antibodies, part of the immune system, targeting oral bacteria, including decay-causing ones.
So cleanser, antibacterial agent, immune defender, all in one.
It is. The source strongly emphasizes. Without salivation, oral tissues get ulcerated, infected, and tooth decay runs rampant. Shows how essential that continuous flow is.
That clinical link really highlights its importance. How is all this regulated? Figure 65-3 shows the nerve pathways.
Salivary glands are controlled almost entirely by the parasympathetic nervous system. Signals originate from the superior and inferior salivatory nuclei in the brainstem near the medulla pons junction.
And what excites these nuclei to boost saliva?
Both taste and tactile stimuli from mouth and pharynx are strong activators. Many tastes work, but sour taste, acids, is especially potent, increasing secretion 8 to 20 times basal rate. Tactile stimuli, smooth objects, cause marked salivation, rough objects, less or even inhibition.
Can signals from higher brain centers influence this, like just thinking about food?
Definitely. Salivation can be stimulated or inhibited by signals from higher centers. Smelling or thinking about favorite foods increases salivation compared to disliked foods. This involves cortex areas for taste, smell, and appetite centers in the amygdala hypothalamus. The source also mentions reflexes from the stomach and upper intestine. Irritating foods or nausea can trigger salivation. Swallowing that saliva helps dilute or neutralize the irritant down below.
And sympathetic input, we heard its dual effect generally. How about for saliva specifically?
Sympathetic stimulation can cause a slight increase in salivation, much less than parasympathetic. Tends to produce thicker saliva. Nerves come from superior cervical ganglia, travel along blood vessels. Finally, blood supply is crucial. Copious salivation needs nutrients. Parasympathetic signals dilate blood vessels. Salivation itself causes more vasodilation. Part of this is due to kallikrein, an enzyme from activated salivary cells, forming bradykinin, a strong vasodilator from a blood protein.
So nerves are primary triggers, integrating senses, brain, gut feedback. Blood flow is essential support, also regulated. Moving down, the esophagus seems simpler secretion-wise.
Esophageal secretions are indeed entirely mucus. Main role, really the only role, is lubrication for swallowing, allowing food to pass smoothly without damage.
What kind of glands make this mucus?
The main esophagus body has many simple mucus glands, like basic goblet cells, continuous lubrication. Additionally, at the gastric end near the stomach, and somewhat in the initial part near the mouth, there are compound mucus glands. Mucus from upper ones protects against abrasion by newly swallowed food. Those near the stomach junction secrete alkaline mucus, protecting against acid reflux from the stomach.
A first line of defense against heartburn, right at the border.
Precisely. A vital protective barrier. Though, as the source notes, despite this, peptic ulcers can still happen there if reflux is severe or chronic, overwhelming the mucus.
All right, into the stomach. A major player, potent secretions. What are the main tubular glands unique to the stomach beyond surface mucus cells?
Besides the surface mucus layer covering everything, the stomach mucosa has two important tubular gland types: oxyntic glands, or gastric glands, and pyloric glands. Oxyntic glands are in the body, and fundus, the upper 80% of the stomach, they're the acid factories, secrete hydrochloric acid, pepsinogen, inactive pepsin, intrinsic factor for B12, and some mucus. Pyloric glands are in the antrum, the lower 20% near the exit. They mainly secrete mucus to protect the pyloric lining from acid. Crucially, they also secrete the hormone gastrin.
Let's focus on oxyntic glands first, the acid and pepsin producers. Source lists the main cell types there.
Yes, the oxyntic gland has different specialized cells, three main ones. Mucus neck cells near the opening secrete mucus. Peptic or chief cells, deeper, secrete lots of pepsinogen. Parietal or oxyntic cells, also deeper, secrete both hydrochloric acid and intrinsic factor. The source also mentions enterochromaffin-like ECL cells secreting histamine, found within these glands too.
Parietal cells, acid producers. Source gets detail on how they make HCl, achieving that incredibly low pH.
It's an amazing, energy-hungry process. When stimulated, parietal cells secrete acid solution, nearly isotonic, but around 160 millimole HCl, pH about 0.8. That's 3 million times the H+ concentration of blood. Needs over 1,500 calories of energy per liter.
Wow, that's a powerful pump.
It is. As they pump H++, bicarbonate goes into the blood, so gastric venous blood is actually more alkaline during acid secretion. Figure 65-5 shows the parietal cell structure, those deep branching canaliculi where acid forms.
Then figure 65-6 details the chemical mechanism, key steps.
Okay, simplified. Water inside splits to H+ and OH. H+ is actively pumped into the canaliculus by the H+ DOS-ATPase pump, the main engine, exchanging for K+ OH. OH combines with CO2 via carbonic anhydrase to form HCO3. This HCO3 exits the cell on the blood side, exchanged for Cl, which enters. The Cl then follows H+ into the canaliculus lumen through channels, forms HCl. Water follows by osmosis. Net result, secretion of water, HCl, some KCl, tiny bit of NaCl.
Incredibly detailed cellular engine. And preventing this super strong acid from leaking back is crucial, right? The gastric barrier.
Absolutely critical. Main components: that thick alkaline mucus layer and very tight junctions between epithelial cells preventing leaks between them. If this barrier is damaged, excessive aspirin, alcohol, H. pylori infection acid leaks back, causing inflammation, gastritis, leading to peptic ulcers.
What signals stimulate these parietal cells to pump acid? Three main factors.
Acetylcholine from parasympathetic nerves, gastrin, hormone, and histamine, local chemical messenger. Acetylcholine stimulates pepsinogen, HCl, and mucus. Gastrin and histamine, however, are primarily and very strongly stimulating acid secretion by parietal cells. Little effect on the others.
And histamine's role is tied closely to those ECL cells in the oxyntic glands.
Yes, key players. ECL cells are nestled deep, close to parietal cells. They make and secrete histamine. The rate of HCl secretion by parietal cells is strongly related to the amount of histamine secreted by nearby ECL cells. Histamine is the direct, potent trigger for the acid pumps.
So histamine is the direct go signal for the parietal cell. But what tells the ECL cell to release histamine?
That's primarily the hormone gastrin. Gastrin is produced by G cells, mainly in the pyloric glands in the stomach's antral portion.
And what stimulates gastrin release from G cells?
Proteins in the food reaching the antrum. Specifically, partially digested protein fragments, proteoses, peptones. That's a special stimulus. This causes G cells to release gastrin into the blood. Stomach churning helps transport gastrin rapidly to ECL cells in the stomach body. Gastrin stimulates ECL cells to release histamine. Histamine quickly stimulates parietal cells. HCl secretion increases, so the chain. Protein in antrum G cells release gastrin. Gastrin stimulates ECL cells. ECL cells release histamine. Histamine stimulates parietal cells. Acid secretion links protein presence to acid needed for its digestion.
Clear command chain. Let's talk pepsinogen. Secreted by peptic cells, mucus neck cells.
Yeah.
Inactive form, right?
Correct. Secreted as inactive pepsinogen. Molecular weight, 42.5 kilonation. As soon as it hits hydrochloric acid, it's activated. Acid splits off a piece, forming active pepsin, 35 kiloday. Pepsin is a proteolytic enzyme, digests protein, but crucially only works in high acidity, optimum pH 1.8 DAC 3.5. Above pH 5, almost no activity, quickly inactivated. So HCl is just as essential as pepsinogen for stomach protein digestion. Activates it and provides the right environment.
And one more vital secretion from parietal cells besides acid.
intrinsic factor, essential for vitamin B12 absorption later in the ileum, secreted along with HCl by parietal cells, critical clinical point from the source. If parietal cells are destroyed, e.g. chronic gastritis, you lose both acid, achlorhydria, and intrinsic factor. Leads to pernicious anemia because B12 can't be absorbed, impairing red blood cell formation, links stomach health to blood count.
Let's touch on the pyloric glands and surface mucus cells, different from oxyntic glands. Pyloric glands in the antrum. What else? And the surface cells covering everything.
Pyloric glands in the antrum are structurally similar but have few peptic parietal cells, mostly mucus cells, identical to mucous neck cells in oxyntic glands. They secrete a little pepsinogen, but primarily lots of thin mucus. Lubricates food movement, protects pyloric mucosa from acid pepsin, and importantly, home of the G cells secreting gastrin, that key hormone.
And the surface mucus cells cover the entire surface between the glands.
Yes, a continuous layer. These surface cells secrete large quantities of viscid mucus, thick, sticky, forms a gel layer, often over a millimeter thick. This is a major protective shell, big part of the gastric barrier. This mucus is also alkaline, lots of bicarbonate, neutralizes acid, trying to penetrate, protecting the wall underneath. Any contact with food or irritation stimulates these cells directly to secrete even more thick alkaline mucus, strengthens the barrier on demand.
Multiple mucus layers.
Yeah.
Thin from neck pyloric, thick alkaline from surface. Smart protection. Let's revisit acid stimulation coordinated with meals. 3 phases, figure 65-7 shows them.
Yes, cephalic, gastric, and intestinal phases. Timing and triggers relative to a meal. Cephalic phase, before food enters the stomach. Seeing, smelling, thinking about, tasting food, more appetite, stronger stimulus. Brain signals, cortex, appetite centers, travel via vagus nerves to stomach. Accounts for about 30% of meal-related gastric secretion.
Just anticipation gets juices flowing significantly. Preparation.
Exactly. Gastric phase, largest when food is in the stomach. About 60% of total meal secretion, bulk of the 1,500 milliliter daily juice. Stimuli, physical distension by food, chemical presence of protein breakdown products. These activate long vagovagal reflexes, stomach to brain and back, local enteric reflexes, and that gastrin mechanism, protein set gastrin release. Lasts several hours while food is present.
And the intestinal phase, final stage.
Occurs when chyme enters the upper small intestine, duodenum mainly. Causes continued, though smaller, stomach secretion, about 10% of acid response, probably partly due to small amounts of gastrin released by duodenal mucosa itself.
This next bit seems counterintuitive. Source says while intestinal phase starts with stimulation, overall, food in the small intestine often inhibits gastric secretion later. Why turn it off?
Crucial feedback. Regulates chyme flow from stomach to intestine. Small intestine can only handle food at a controlled rate. Too fast overwhelms digestion absorption. This inhibition helps slow the stomach. Triggered by at least 2 influences from the small intestine. First, the reverse enterogastric reflex. Food in small intestine initiates nerve reflexes, enteric, sympathetic, vagus, back to the stomach, inhibiting secretion and motility. Triggered by distension, acid, protein products, irritation in upper intestine. Key part of slowing stomach emptying when intestine is busy. Second, presence of various things in upper small intestine: acid, fat, protein products, hypoosmotic fluids, irritants causes release of several intestinal hormones.
Which hormones have this inhibitory effect on the stomach?
Secretin is one, known for pancreas effects, but also opposes stomach secretion. Others with slight to moderate inhibitory effects: glucose-dependent insulinotropic peptide, GIP, or gastric inhibitory peptide, vasoactive intestinal polypeptide, VIP, and somatostatin. Purpose of these inhibitory factors, nerve and hormone, is clearly to slow down chyme delivery when the small intestine is already processing, overloaded, or irritated. Acts as a brake.
Sophisticated feedback for downstream efficiency. What about between meals, the interdigestive period? Stomach completely quiet.
No, not totally silent. Still secretes a few milliliters of gastric juice per hour, usually almost entirely non-oxyntic type, mainly mucus, little pepsin, almost no acid. Baseline protection. But fascinating link. Emotional stimuli can significantly increase this interdigestive secretion. Makes it highly peptic and acidic, like cephalic phase, up to 50 milliliters or more. This increase from emotional stress is thought to contribute to peptic ulcers. Constant unnecessary acid might overwhelm protective mucus.
A powerful connection between emotion and gut physiology with real consequences. Source briefly touches on chemistry of key GI hormones. Gastrin, CCK, secretin. What's noted?
Notes they're all large polypeptides. Gastrin MW2000, CCK 4200, secretin 3400. Interesting detail. Gastrin and CCK share the same terminal 5 amino acids.
And that shared part is functionally important.
Yes, gastrin's activity resides in its terminal 4 amino acids. CCK is in its terminal 8, including the shared bit. For secretin, the whole molecule is needed. Source mentions synthetic pentagastrin, gastrin's terminal 4 plus alanine, mimics all natural gastrin properties. Neat structure-function example.
Great detail. Moving on, the pancreas. Another powerhouse structure. What digestive juice does it contribute?
Pancreas. Large compound gland behind stomach. Structure similar to salivary glands, figure 65-2, a sheeny-feeding ducts. Secretes pancreatic juice into duodenum. Vital for digesting everything, neutralizing acid, digestive enzymes specifically made by pancreatic acini, but the large volumes of sodium bicarbonate solution, acid neutralizer, secreted separately by ductula duct lining cells. Combined juice flows via pancreatic duct, usually joins common bile duct, empties into duodenum via papilla of Vater, controlled by sphincter of Oddi.
When does it secrete most? Continuous or meal-timed?
Most abundantly in response to chyme entering the upper small intestine. Juice characteristics, enzyme mix can vary depending on food type and chyme. Adaptive.
Source carefully distinguishes this from the pancreas's other famous role.
Yes, important distinction. This is exocrine digestive function. Pancreas also has endocrine function. Islets of Langerhans secrete insulin glucagon directly into bloodstream for blood sugar regulation. Separate job.
What are the main components in this vital pancreatic digestive juice? The complete arsenal.
Packed with enzymes for all 3 major food types. Proteins, carbs, fats. Makes it essential for complete digestion. Plus, crucially, large amounts of bicarbonate, HCO3, to neutralize stomach acid.
Let's break down the specific enzymes.
Proteins. Most important are trypsin, chymotrypsin, carboxypolypeptidase. Trypsin most abundant. Trypsin, chymotrypsin break whole partial proteins into smaller peptides. Carboxypolypeptidase splits some peptides into individual amino acids. Carbs. Pancreatic amylase. Continues starch digestion, started by saliva, hydrolyzes starches, glycogen, most other carbs, not cellulose, into disacchardestriate saccharides. Fats, 3 key ones. Pancreatic lipase, most important, breaks neutral fat into fatty acids monoglycerides. Cholesterol esterase, hydrolyzes cholesterol esters. Phospholipase, splits fatty acids from phospholipids.
Truly a complete set, but like pepsinogen, are the powerful protein-digesting ones secreted inactive to protect the pancreas.
Absolutely essential. Must stay inactive until the intestine. Synthesized as inactive zymogens. Trypsinogen, chymotrypsinogen, procarboxypolypeptidase. Trypsinogen activation is key first step. Activated by enterokinase, an enzyme from intestinal mucosa when time touches it. Once a little active trypsin forms, it can activate more trypsinogen, autocatalysis. Positive feedback. Active trypsin then activates the others, chymotrypsinogen and procarboxypolypeptidase.
Why so critical they aren't activated inside the pancreas? The danger.
Literal self-digestion. Active enzymes would digest pancreatic tissue itself. Leads to acute pancreatitis severe, often lethal, fortunately a critical protection. Same cells secreting zymogens also secrete trypsin inhibitor, found in cytoplasm, ducts. Binds trypsin tightly, prevents activity inside pancreas. Since trypsin activates others, this inhibits all proteolytic enzymes there.
And if this fails?
Acute pancreatitis. Injury or blocked duct, secretions, pool inhibitor, overwhelmed enzymes activate, digest pancreas. Severe pain, shock, possible death, or lifelong insufficiency. Shows the precarious balance.
Really underscores that inhibitor's importance. Agini make enzymes. The source says bulk fluid bicarbonate comes from elsewhere.
Yes, water and large amounts of bicarbonate ions, HCO3, secreted mainly by epithelial cells lining pancreatic ductal ducts, not acini. When heavily stimulated, HCO3 concentration in juice can be very high, up to 145 milliQL, about 5 times plasma concentration. This high bicarbonate is essential for neutralizing the large amount of HCl entering duodenum from stomach.
How do duct cells manage this high bicarbonate secretion? Figure 65-8 shows the mechanism.
Sophisticated ion transport. CO2 from blood diffuses into duct cell, combines with water via carbonic anhydrase on carbonic acid, mol splits to H plus and HCO3. More HCO3 can enter from blood side too. HCO3 transported across luminal membrane into duct lumen, often exchanged for Cl, H plus pumped out on blood side. This ion movement, Na plus HCO3, creates osmotic gradient, pulling water into lumen, forms large volume of alkaline fluid.
Coordinated effort using CO2 to make this alkaline flood, and its role neutralizing stomach acid is absolutely critical.
Absolutely essential for survival. Reaction: HCl plus now, HCO3, NaCl plus H2CO3, NaCO2 plus water, CO2 briefed out, leaves neutral salt solution. Rapid neutralization does vital things, stops pepsin activity, needs acid, protects small intestine lining from acid burn, can't handle pH 0.8, prevents duodenal ulcers, creates optimal slightly alkaline environment, pH 7.0 DAOs 8.0 for pancreatic enzymes to work.
Multitasker, protector, pH setter, enzyme enabler. How is this vital pancreatic secretion regulated? 3 basic stimuli.
Primarily controlled by nerves and hormones responding to food. 3 main stimuli. Acetylcholine from vagus enteric nerves, hormone cholecystokinin, CCK, hormone secretin. Acetylcholine and CCK primarily stimulate acinar cells, a large enzyme production, less watery bicarbonate fluid. Enzymes might just sit in ducts without enough fluid. Secretin, contrastingly, primarily stimulates ductal epithelium, large secretion of water solution rich in sodium bicarbonate.
So acetylcholine CCK for enzyme tools, secretin for the alkaline flood, needed to use them in the right environment.
Key functional distinction. Source also notes powerful multiplicative effect. Stimuli together cause much greater secretion than some of individual effects. They potentiate each other. Normal, copious secretion relies on this combined stimulation.
Pancreatic secretion also has phases like gastric.
Yes, cephalic, gastric, intestinal, cephalic anticipation. Vagal signals, acetylcholine, moderate enzyme secretion by Ashini, 20% total enzyme. Gastric, food and stomach. Vagal enteric reflexes continue enzyme stimulation, another 5–10%. But little fluid in these phases, enzymes mostly wait in ducts.
Intestinal phase is where pancreas really kicks in, releases juice into duodenum.
Absolutely. Most copious, most important phase. When chyme enters small intestine, mainly driven by hormones from duodenal jejunal mucosa.
How does secretin, the bicarbonate hormone, work here?
Secretin released from S cells in duodenum jejunal mucosa, triggered most potently by acidic chyme from stomach, pH 4.5 5.0, SPO 3.0. Secretin travels via blood to pancreas, stimulates ductal cells, secrete large volume, high bicarbonate fluid, critical negative feedback. Acid enters secretin release, bicarbonate secreted, acid neutralized, protects duodenum, sets optimal pH for enzymes.
And CCK's role in intestinal phase, the enzyme hormone.
CCK released from eye cells in duodenum upper jejunal mucosa, triggered mainly by protein breakdown products, proteoses, peptones, and long-chain fatty acids in chyme. CCK via blood to pancreas primarily stimulates acinar cells, secrete huge amounts. Liver constantly secretes bile, 600 × 1,000 ml a day. 2 vital functions. First, crucial for fat digestion and absorption. No enzymes itself, but bile acids do 2 key things: emulsify large fat globules into smaller droplets, increase the surface area for lipase, and help absorb digested fat products, fatty acids, monoglycerides, by forming micelles tiny complexes ferrying fats to intestinal wall. Source stresses. Without bile salts, up to 40% ingested fat lost in feces. Second, bile is a major route for liver to excrete waste products from blood into gut for elimination. Bilirubin from old red cells, excess cholesterol.
Source describes bile secretion in 2 stages, figure 6511.
Yes. Stage 1, initial secretion by main liver cells, hepatocytes. Contains bile acids, cholesterol, phospholipids, lecithin, bilirubin. Flows into tiny bile canaliculi between liver cells. Stage 2, as bile flows through bile ducts. Duct lining cells add watery solution rich in sodium and bicarbonate. This secondary secretion can double total volume, stimulated specifically by secretin, adds more bicarbonate to help neutralize duodenal acid.
But most bile doesn't go straight to duodenum, stored first.
Correct. Most stored and concentrated in gallbladder until needed, typically during aftermeal. Gallbladder small, 30–60 milliliters, but lining absorbs water, small electrolytes, concentrating bile salts, cholesterol, lecithin, bilirubin, 5 to 20 times. Stores hours of liver secretion.
What's bile composed of after concentration?
Table 65-2 compares. In concentrated bile, bile salts most abundant solute, half total. Bilirubin, cholesterol, lecithin, also significant, some electrolytes. Concentration removes mostly water, most electrolytes.
What triggers gallbladder to contract empty concentrated bile when food, heparin, fatty arise?
Gallbladder emptying timed with meals, starts 30 minutes after eating, strongest stimulus fatty foods. Mechanism, gallbladder wall contracts rhythmically, sphincter of Oddi at duct exit to duodenum relaxes. Most potent trigger for gallbladder contraction. Hormone CCK. We know CCK released from duodenum by fat protein products. Travels via blood to gallbladder, contracts forcefully. Simultaneously relaxes sphincter of Oddi bile, squirts into duodenum. Acetylcholine, vagal enteric nerves, adds lesser stimulation.
So fatty food CCK release, main signal for gallbladder contraction bile delivery.
That's the key pathway. Emptying poor without fat. With fat, usually empties completely in 1 hour. Figure 6511 summarizes this.
Let's get more detail on how bile salts work in fat digestion absorption. Where do they come from?
Liver cells make 6G bile salts daily from cholesterol. Cholesterol converted to primary bile acids, salicylic, chenodeoxycholic, then conjugated with glycine or taurine conjugated bile acids, secreted as sodium salts into bile. Two vital actions in intestine. First, detergent action. Emulsify fat globules, break into smaller droplets for enzymes. Second, more important, is source. Form micelles tiny complexes with digested fats, fatty acids, monoglycerides. Micelles ferry these fats through watery gut contents to intestinal lining for absorption. Without micelles, poor absorption, fat loss.
And the enterohepatic circulation. Bile salts highly recycled, not just used once.
Exactly. About 94% reabsorbed from small intestine, half by diffusion early, rest active transport in distal ileum. Enter portal blood, return to liver, absorbed by liver cells, resecreted into bile. This cycle, enterohepatic circulation, means same salts cycle 17 times before loss in feces. Liver constantly makes new ones to replace small daily loss.
So liver production linked to recycling.
Yes. Liver bile secretion rate depends heavily on bile salts returning via enterohepatic circulation. More returning salts, more secretion. Source notes. If salts lost externally, fistula, liver ramps up production 6-10-fold to compensate.
And secretin increases bile secretion too.
Yes. Besides strong stimulus from bile acids, secretin, triggered by duodenal acid, also increases bile secretion, sometimes doubling it. But this is mainly the watery bicarbonate-rich secretion from bile ducts, not increased bile salt production by main liver cells. Adds more bicarbonate to help neutralize acid.
You mentioned cholesterol excretion via bile. Source links this to gallstones.
Liver cells secrete 1,2-G cholesterol into bile daily, synthesized from body cholesterol. Cholesterol insoluble in water. Bile salts lecithin in bile-form micelles, keeping cholesterol soluble. When bile concentrated in gallbladder, these components concentrate together, usually maintaining solubility.
But if balance is off, cholesterol precipitates.
Yes. If cholesterol exceeds solubilizing capacity of bile salts lecithin, it precipitates cholesterol gallstones, figure 6512. High dietary fat obesity, increased cholesterol secretion, raise risk. Also, gallbladder lining inflammation can alter absorption, absorbs too much water bile salts. Cholesterol concentrates excessively, precipitates gallstones form.
Imbalance or gallbladder dysfunction, weighs stones. Moving down, small intestine's own secretions, key contributions.
First part of duodenum, first few centimeter, has unique Brunner's glands, compound mucus glands secreting large amounts of alkaline mucus.
What triggers them?
Tactile or irritating stimuli on duodenal lining, like acidic chyme, strongly stimulate. Vagal stimulation, hormones, SP secretin, also increase secretion.
Their function.
Primarily protection. Alkaline mucus protects duodenal wall from acidic gastric juice. Adds bicarbonate to help neutralize alongside pancreatic biliary. Source notes. Sympathetic stimulation inhibits these glands. In stressed people, might leave duodenal bulb unprotected, contributing to its high ulcer frequency.
Besides Brunner's glands, entire small intestine surface has crypts of Lieberkühn between villi. What do they secrete?
Crypts, figure 6513, are pits between villi. Lining has goblet cells, mucus, and enterocytes. Enterocytes in crypts secrete 1,800 milliliter daily water electrolytes, basically pure extracellular fluid, slightly alkaline, pH 7.5, 8.0. Enterocytes on villi primarily reabsorb this fluid, creates flow from crypts to villi, watery vehicle helping digestive products contact villi for absorption.
How do cryptenterocytes secrete that watery fluid?
Exact mechanism unclear, but involves active secretion of chloride and bicarbonate into lumen, sodium follows, osmotic gradient pulls water in.
Source mentions small intestine digestive enzymes, but says almost none in collected fluid. Confusing if digestion happens there.
Really important point. Major small intestinal digestive enzymes aren't secreted into lumen in bulk. Located within villi enterocytes, embedded in brush border membrane, they do terminal digestion as substances are absorbed through cell surface.
Like enzymes built into the absorptive surface.
Essentially, examples, peptidases split small peptides, burdensome amino acids. 4 disaccharidases, sucrose, maltase, isomaltase, lactase, split disaccharides, monosaccharides, some intestinal lipase. Source notes, rapid cell turnover. Crypt cells divide, migrate up villi, express enzymes, shed every 5 days. Releases some enzymes into lumen, but primary action is cell surface.
Rapid turnover, amazing for absorption repair. How is small intestine secretion regulated?
Most important regulation for crypt secretion. Local enteric nervous reflexes, mainly triggered by physical presence irritation of chyme, largely local self-regulatory.
Finally, large intestine. What secretions does it contribute?
Large intestine also has crypts of Lieberkühn, but no villi. Epithelial cells secrete almost no digestive enzymes, mostly mucus cells secreting only mucus. Contains moderate bicarbonate, alkaline, pH 8.0.
How is large intestine mucus secretion controlled?
Primarily direct tactile stimulation from fecal contents, local nervous reflexes to mucus cells in crypts. Parasympathetic stimulation, pelvic nerves, also increases mucus secretion, SB distally, often dramatically. Source example: severe emotional stress, intense parasympathetic stimulation, copious mucus secretion, frequent mucus-only bowel movements.
What functions does this mucus serve in large intestine? Main job: water absorption, feces formation.
Protects wall from abrasive damage by solidifying feces. Helps bind fecal matter together. Protects wall from extensive bacterial activity. With alkalinity, helps neutralize acids from bacterial fermentation.
Source links excessive secretion here to diarrhea.
Yes. Intense irritation, e.g. bacterial infection enteritis. Mucosa secretes extra large amounts water electrolytes plus mucus. Dilutes irritants, speeds transit, diarrhea, fluid loss. Problematic, but helps wash away irritants, potentially speeds recovery.
Wow, we really went deep there. From tiny goblet cells to the complex hormonal and nervous symphony controlling the pancreas and liver. Just an incredibly precise and vital system.
It truly is. Understanding the different secretions, their specific jobs, digestion, lubrication, protection, neutralization, and the intricate controls reveals the remarkable coordination needed for proper digestion and really overall health.
We cover the details. Saliva's composition and regulation, the stomach's multi-layered defenses and potent acid system, the pancreas's critical neutralizing and enzymatic power, bile's vital role in fats and waste. Not to mention the intestine's protective mucus and preparatory secretions.
And how disruptions from lack of saliva to overwhelmed trypsin inhibitor or insufficient bicarbonate can lead to significant health issues. Highlights how interconnected it all is.
So you've now got the lowdown, the deep understanding of the secretory engine of your gut, straight from the source material.
Think about it. Next time you eat, that simple act triggers this cascade of precisely tailored secretions from the moment you see food to hours later. It's all happening.
It's pretty mind-blowing when you consider the level of detail involved. That wraps up this deep dive. We hope this has given you a solid foundation and some real aha moments about this crucial physiological system.
Keep exploring, keep questioning, and keep that curiosity alive.
We'll catch you next time for another deep dive.